there's nobody else on at this point.
No, but they catch up later, so that's all good.
Okay.
So hello, everyone. This is a bit in-house for people that are on the call right now, but hopefully if people catch up, as we were saying afterwards, welcome. Sorry that this is a reschedule of the last of the four of the Lunch and Learn PacBio sessions. We had some technical problems last week, so this is the reschedule from the 11th now on to the 18th of May. So I shall share my screen. And why are you being unpleasant to me? Okay. All right. So as I said, this is the PacBio Lunch and Learn series of webinars have run over 8 weeks. There's been 4 sessions in all, a fortnight apart. We started off with an overview of how PacBio sequencing, how HiFi reads are generated on a PacBio sequencing. HiFi reads are these very long reads, very highly accurate reads. And we went through some of the basics of how that happens, how you make the library, and how the sequencing machine runs round and round the molecule and reads it multiple passes to get these very highly accurate consensus reads, which is HiFi sequencing. And then we sort of started to talk about a few applications. So the second webinar, we talked about microbiome, about how to look at either 16S or total DNA from your microbiome samples, whether that be soil or swabs from human nasal swabs and such like, that sort of thing. So that was session 2. And then session 3, we talked, I suppose, about the general whole genome sequencing, so plant and animal genome sequencing, and the power that you have by having very long reads, but also very highly accurate long reads, and how that makes the computing so much faster and easier to make those assemblies when you have that data available. So today, not the 11th of May, as I said, this is a reschedule from last week, but we're going to talk about transcriptomics, so sequencing the transcriptome from your experimental cells or organisms, whatever, and how this can be done, and the power again of having that very long read and very highly accurate read, and why this is important when you're looking at the transcriptome. So there we go, HiFi for transcriptomics. Okay, so running through this, I'm just going to move this photo over so I can see my screen better. What PacBio called transcriptome sequencing is ISO-SEQ, because ISO for isoforms, and so they're sequencing full-length messenger RNA, and they're able to pull out all the isoforms that are there, which is again, we'll go into this in a little bit more detail, but as the diagram on the right sort of shows, this is quite a difficult thing to do with short read sequencing, because when you sequence short reads, the chances of actually getting a short read that bridges the intron-exon junctions, those splice junctions, splice sites, is quite rare. So you only get a very small subset of your short reads that enable you to link the different exons together, and therefore, you have a lot of trouble finding all the different splice variants that are available in your sample. So you don't find all the isoforms very easy. It's very difficult to link it all together. But you have the power to sequence full length and discover all the isoforms that are available. And because it's single molecule sequencing too, you're essentially getting a digital count as well of the expression differences between the different isoforms. So if something's at 90% of all the isoforms compared to something else at only 10%, then you see those differences in sequencing terms too. So now I've talked a little bit about the diagram up there. I suppose I'll just run through the notes here. So ISO-SEQ is full-length cDNA sequencing, so there's no assembly required. You get absolutely all the information. It can be targeted if you've got a capture system, or you can do whole transcriptome, entirely up to you. And the power of it, as I said, is because you see everything, you see all the splice variants, you can discover previously unknown splice variants and isoforms that haven't been identified before. It also, from that, helps with genome annotation, and because of the power of the sequencing, you can do this with or without a reference genome. So you're not just tied to model organisms either. And of course, it increases the accuracy of RNA-seq and the level of resolution that you can find. So, and we'll go into that in a little bit more detail as we go through the talk. But the great thing about it is that you start here with messenger RNA and you make cDNA, and from that point, really, the protocol is essentially identical as if you were sequencing any other DNA on a sequel instrument, sequel or sequel 2 or sequel 2e, as there is now. The library prep is really much the same once you've ended up with cDNA. And you're able to then sequence or even multiplex onto the SMRT cells. The great thing is too that the bioinformatics is kind of being handled for you as well. There's pipelines available already programmed in SmartLink to handle the analysis, to essentially tidy up all those isoform reads and map them back if you wish, and do a count and tell you what isoforms are available for each gene. So fortunately, with all of these things these days, there's a kit for that. So PacBio provides the ISO-SEQ Express kit for doing this type of transcriptomic sequencing. So you want to start off with total RNA from your tissue, whatever that be, whether it's plant or animal or whatever. And you need about 300 nanograms total RNA. And from there, you're going to run a reverse transcription to make your cDNA, and then you can amplify that, and then you've got options to multiplex and put multiple samples together on one SMRT cell, and then you go on to your sequencing. And from the sequel 2 or 2e system, where you've got the SMRT cell 8Ms, you're going to generate up to about 4 million full-length reads on that SMRT cell. So of course there's a protocol. All the PacBio's protocols have this procedure and checklist as their start, and so this is the one you'd need for ISO-SEQ. It's the ISO-SEQ Express template prep for sequel and sequel 2 systems, and it goes through all the details of everything you need, and it's about a one-day protocol to go through to start with your RNA and go through to the sequencing, and yeah, it tells you how to QC your RNA, because obviously that's important. You need to get good quality RNA going in. So it tells you how to best ways recommended for QCs, and then for performing your cDNA synthesis and making those all important SmartBell libraries. That's the library sequence with the SmartBell adapters that make it essentially a circular molecule, so you can sequence round and round and round and get these very highly accurate HiFi reads. So it works in combination with a kit from New England Biolabs, NEB, the NEB Next SMRT Cell Low Input cDNA Synthesis and Amplification module or kit. And there's a bit of a sort of diagram here of essentially what's going on. So there's a combination of primers that come with the PacBio ISO-SEQ kit and also with the NEB kit here. So you're going to start up here with your total RNA. And of course, you're interested in the transcriptome, so you're particularly interested in the stuff that's got a poly-A sequence on the ends. And that gives you a handle, doesn't it, to get hold of. So you can bind an oligo-DT primer to the poly-A, and you can run your first strand synthesis. Now, the reverse transcriptase enzyme that runs this very conveniently for you tends to put 3 Cs on the end of the molecule. That's what it does, and that again gives you another handle now at the 5' end of the molecule. So then you can use your so-called template switch oligo, which is in the PacBio kit. So the yellow and orange are all in the PacBio kit. The blue and teal color are in the New England Biolabs kit. But you can bind your template switch oligo onto the 3 Cs here by using a primer that has 3 Gs on the end. So that allows you to sequence back through the second strand to make your double-stranded cDNA. Because you've now got handles on each end, you know you've got some sequence here from the reverse transcription primer and from the template switch oligo. You can now amplify this up to make your nicely amplified library. So that's really the way you go about making this cDNA ready for your sequencing. And then for those of you that were on the calls before, you'll recognize the topology of these molecules, of these SMRTbell molecules. So you're able to then take your full-length cDNA that you have and bind on these SMRTbell adapters to make these circular molecules. And of course, for this, you've got some options of exactly which way you do this. You can either grab the cDNA and put on the sort of standard SMRTbell adapters and sequence away. You could have a 3' bar-coded adapter just on that 3' end if you wanted to multiplex a low-plex, or of course you can go for double bar-coded, so you can have bar-code at 3 and 5' end if you're wanting to multiplex more heavily or gain more information that way. Or of course if you've targeted the cDNA, so if you've done some sort of targeted capture, hybridization capture, targeted genes, you can do that, which will focus more on genes of interest out of the total transcriptome. So you've got options of how you do this. There's a little chart of showing the power, I suppose, of being able to sequence the full-length molecule very accurately. The example here in black gives you the reference genome sequence. So the black boxes here are the exons, and the left lines joined together are the introns. So when you actually look for your transcriptome sequences for all the isoforms, you can see that you get one that is exactly like the reference, effectively a full splice match, as it's called. But alternatively too, you get a lot of other things. So you're getting all the alternatively spliced, so-called incomplete splice matches, and some novel splice matches that may have been recognized before, so they're in catalog, but it's also finding a lot of completely novel isoforms that hadn't so far been characterized. And this is just an example of the number of reads and compared to the number of detected isoforms that you see, which just demonstrates the power and accuracy of the HiFi sequencing and what you can do with that kind of data. So this was on one of the first slides, just in a little bit more detail now, so you can see it a little bit better. But again, you've got your gene sequence, and here you're getting the various splice isoforms, and it's just demonstrating what I said at the beginning of how difficult this is to do with just short read sequencing, because only the ones here with the red are the subset of short reads that actually are in these splice junctions. So it allows you to join these exons together and know what's there. But it's very, very difficult if you say skip one exon out and you find a splice junction between, say, exon 2 and 4, and exon 3 is missing. But then what's the rest of the molecule? How do you know where it all fits together when you've only got short reads? It's very, very difficult to do, and it's much, much easier when you have the full length molecule sort of digitally counted across for everything. So it really does make a difference. During the library prep part, of course, you've made your cDNA, and you end up through the protocol of doing some magnetic bead cleanup steps. This is noteworthy, I think. Use Pronex beads with the ISO-SEQ protocol, and you can very slightly target the size range of the cDNA you want to capture at this point, if you wish. You can go for that kind of mode around 2 kb, which is unless you have some a priori knowledge, is probably a good place to start. It's sort of marked in green here. It's this middle peak. So capturing, your average gene, if there's such a thing, of around 2 kb is probably a good start point. But if you have certain knowledge message that that your gene of interest is long, or if you're after particularly short molecules that might otherwise be missed, you can slightly adjust your Pronex bead ratios to capture either the small molecules below 2 kb, or to capture the larger targets above 3 kb, as shown in the sort of blue and the pink here, respectively. So it's an optional point in the protocol, and I think it's worth noting if anybody was thinking, well, I know that that my gene is quite long. You know, if you're capturing the average, will I miss my target? Well, you can actually bias it a little bit in your favor if you wish. So I suppose this is the nuts and bolts of it, and we'll go through some diagrams here to show really what's your sequencing, and then how you're analyzing that sequencing. But this is the key take home, I suppose. The ISO-SEQ analysis transformed HiFi reads into high quality full length isoforms, and so you have this mismatch of sequencing data that is all your different transcripts, and the software pipeline is able to show you which isoforms you have for the different genes in your species that you're looking at. So we'll run through this sort of slowly. There's in SmartLink 9, which is actually SmartLink 10.1 has just come out for the Sequel 2E instruments. So this is just very slightly out of date, but it's essentially the same with SmartLink 10.1. But with SmartLink 9, you have 3 options of the way you run the analysis. You can either run the standard, or you can run without clustering, which is a little bit shorter, or you can do it with full mapping, which is a little bit longer. So you've got the 3 options available. So to go through, you're going to take these CCS reads, or HiFi reads as they are, where you've sequenced around the molecule many times, so you've got very, very highly accurate consensus sequence. You're going to put in perhaps a demultiplexing step if you've put in multiple samples and barcoded. So this software will look for the barcode, take those CSIRO reads, look for the barcoding, and demultiplex at that point, and then refine. And we'll talk about these various steps as we go through. But it's then going to refine those sequence reads. You know, chop off primer sequence and tidy up all the reads before it does anything else. The normal pipeline, I suppose, then includes the clustering step. So it's going to look for the various isoforms and look for the ones that are the same, and cluster those together and work out which isoforms go to for which gene. And then that goes to the mapping. If you're going to collapse all of those different isoform reads and map them all back to the transcriptome, that gives you the full pipeline that's available. So going through, you start off with your consensus sequencing reads, the CCS reads here, the HiFi reads, as we saw before. So you've got all the different reads. You've got some options on the way you decide if a read is passed into this so-called CCS or HiFi mode. Recommend for the ISO-SEQ protocol, and this is all in the guides, that you put the polish option to on, and that you set for a Q20 a minimum of 3 passes around the molecule. So if you remember here, your polymerase binds to the recognition site here with the sequencing primer to form a complex, and then it sequences around the molecule as it churns around, like as a sort of bicycle chain going through the cogs, if you like. And so you're going to sequence, say, the purple piece here. It's going to go around the smart bell, and then going to sequence the opposite strand in yellow, background the smart bell purple. It needs to do at least 3, so it needs to go at least purple, yellow, purple to get minimum 3 passes. But hopefully with your sequencing run and the movie time that you set on the sequencer, you'll get multiple passes, more than 3. You also require probably setting the minimum RQ at 0.9. Okay, so this again just demonstrates where you get random polymerase errors in the sequence. You're able to effectively screen those out when you form the consensus, because you'll have multiple passes over that base, and it will call the consensus read and get rid of any random polymerase errors. So then to demultiplexing, a fairly straightforward thing in a bioinformatics pipeline. You're going to take those CCS reads. It's going to have a look at those and look for the barcodes and demultiplex those according to your various samples that you've mixed together. It's important to note that you should in the ISO-SEQ pipeline demultiplex with an algorithm from Lima. Which has a special ISO-SEQ mode for it, and it will sort out the 5' and 3' primers and remove those, and don't use other demultiplexing applications because they may well not be able to do a very good job or chop off bits of the sequence that you need. So it's important to use the Lima algorithm when you're demultiplexing for ISO-SEQ. So now some terms here, I suppose. You end up with nicely trimmed and demultiplexed full-length reads. So you can see you've got the cDNA with its poly A on the end, and you also have a few spurious ones in there, of course. So the refine step of the pathway basically takes those demultiplexed HiFi read files that are full length. And that have the primer file and removes concatenamers to form these so-called FLNC reads, full-length non-concatenamer reads. Okay, so if your sample has poly A, you require a poly A step to filter to look for those full-length reads. So make sure that you've gone through to the poly A tail. And there's some other parameters there to make sure that they are good quality reads. And you end up with a bucket then of these full-length non-concatenamer reads, so all the reads tidied up. So then the clustering step takes these so-called FLNC reads, full-length non-concatenamer reads, and it begins to cluster them into the various isoforms, which enables you to see all the variability that's happening at the transcriptional level. Again, you need very high quality reads, so greater than 99%, and yeah, it puts them into the two buckets. So it also it doesn't delete, but it also marks these as high or low quality reads. So once you have these clustered isoforms, if they're in the CCS or HiFi mode, it's fine. They don't really need to be polished too much, but there is a polish step available if you've done these continuous long read type sequencing, which you probably won't be doing on the Sequel II and Sequel IIes now, and even on the Sequel instrument, you'd probably be looking at the read lengths getting so good and everything being so stable that the reads are long enough. You don't need to go for the CLR polish of those reads. You'll have CCS sequence, so it's all very accurate anyway. But it's important that in order for these isoforms to be considered the same, there's a set of rules effectively that are built into the pipeline. If you need the 5' start needs to be less than 100 base pairs different from one another. If it's under 100 base pairs, it will still put it into the box that they're the same. Less than 30 base pairs difference at the 3' end. Again, if there's under 30, it will put them in the same basket. And of course, less than 10 base pair internal gaps. Though there is no limit on the number of gaps, you can have multiple gaps as long as they're less than 10 base pairs anyway, then it will put them into those and consider them the same isoform. And then finally, if you're wanting to go to this step, if you have a reference genome or whatever, you can map and collapse these reads. So now you can take these very highly accurate, full-length polished isoforms, designated here with the colors, and then map back to your reference genome using Minimap2, and that's where it starts to show you for a particular gene the different isoforms that are available, and so how it's done its splicing and made the various full-length cDNA. Yeah, full-length messenger RNA, which you've seen from your cDNA. So that's essentially how the pipeline runs once you've made your libraries and run your sequencing. So a little bit of data here, a number of different runs here just to show the kind of accuracy and reads that you get using ISO-SEQ. This is run on a Sequel II system under the recommended guidelines. So it's a 24-hour collection time for the movie, looking for the sequence incorporation. And there's a 2-hour pre-extension time, which allows the molecule to stabilize and get into this very stable rolling circle sort of confirmation for sequencing that goes into HiFi. So here, what's been sequenced here is a universal human reference RNA sample. So it's a sort of synthetic sample, if you like, that's been sequenced here multiple times as an example. But just going through, there's eight different sets of sequencing here that's done, so 8 data sets. You're looking at an average of, high 200 to 288 gigs of data on average from each run. So certainly most of them over 250, some of them up over 300 gigs of data per run. And the number, as we said, you'd be estimating around about 4 million CCS, or very high quality long HiFi reads per sample. When these get filtered, as you see, the filtering removes some of those reads, but not too many. So you've still got over 3.5 million on average at that point. So you're talking about 80 to 85% of those reads passing filter, and the length here of those reads being around about 3.5 kb. So that's from a sort of set of, if you like, a synthetic human RNA sample. But here, I suppose, but more real life, that human reference sample is included here. But then there's some other typical sort of tissue samples that you might be looking for transcriptional information here, where it doesn't give organism as human, but otherwise it's mouse here. Again, the protocol that I alluded to, where you can adjust your pronex speed ratios a little bit, is the standard is capturing around that mode of 2 kb. The long is capturing those slightly longer molecules up to maybe 4 or 5 kb, and the short is the stuff less than 2 kb. So again, this was run on these different samples. You're still looking at around 3 or whatever odd million of these filtered non-concatenized reads per sample, and again 80–85% whatever for the percent of reads that you get through. Just note that when you do the shorter molecules, you just notice that the number of filtered reads you get here is a little bit less than the average, but that's just part of going for the shorter sequences. So a few examples of that have been published out there, and then we'll go into a little bit more detail on an example. But things that people have done using ISO-SEQ. Well, there's quite a lot being used in agricultural samples. I've tried to. There's many obviously in human and mouse and model systems as well. I've tried to sort of stray away from that in these examples and look at things away from model organisms. So here in agricultural sciences, people looking at crosses between, in this case, different cattle. So you've got your Brahman versus Angus cross, and looking at the isoform in those progeny. So looking specifically for the different isoform and expression, and which alleles have come from which parent, and also looking at tissue specificity, so to see particularly the transcriptome in say muscle tissue against and milk production in some cases, if it's for milking cows rather than beef cows, as you see here. So those sorts of things that have been done and have been published. Slightly interesting example here. Cannabis has been analysed specifically for its active ingredients, so for its THC and for its CBD synthesis. That's been looked at, and I believe things like poppies and other things for where there's medicinal compounds that are being used. There has also been analysed, and also in the cannabis publication here, they looked at the Y chromosome gene annotation there as well. Slightly off the wall one, I don't know who on earth went and collected samples from this, so they were good luck to them for that one. But grizzly bear, of all things, they've run ISO-SEQ on grizzly bear samples and comparing bears that were active against those that were hibernating to see transcriptional profile differences, and also looking, because there was no information, at alternative splicing that was going on in bear samples. So yeah, there's lots and lots of wide and varied applications for ISO-SEQ is, I suppose, the point I'm trying to make with the slide here. So in a little bit more detail, this is, I guess, a plant example of a bit like the cattle example above, but just in a little bit more detail. I don't expect you to be able to see this in too much detail, but it gives you an idea. There's two elite maize lines that were in part of a breeding program. And the researchers were particularly interested in two dominant isoforms. This is so-called PB 8517.4 and 8517.1, and there's some specific information about those. So the isoform 0.4 has 11 exons, but the isoform 0.1 has the last exon spliced. Okay, and from the breeding trial here, parent B73 only expresses the 0.4, the fully unspliced version, and KI11 only expresses 0.1, with the missing 3' exon. So they set up and did quite a scholarly job here, I thought. They set up the crosses both ways, if you like. So they used B73 as the male parent in one cross and the female parent in the other. So so you have the full gamut, the full subset. And again, once you examine this with ISO-SEQ, you can see the mixture across the progeny of these crosses, containing both isoforms, the 0.1 and the 0.4 isoforms, inherited either way from male and female parent, whichever way that they were crossed from the elite lines. So you get a lot of power, and you see exactly the splice junctions and exactly any differences within the isoforms and how they're being expressed across all the progeny of this cross. So very powerful. So I suppose rounding up a little bit with this, and we do have a little bit to talk about just after this, a different application that's related to ISO-SEQ, something a bit more specific, if you like, but the best practices for ISO-SEQ here are just summarized. For making your libraries, you want to make your full-length cDNA from about 300 nanograms of total RNA. As we said, good quality. There's good QC checks or check out that you've got good quality RNA. The PacBio ISO-SEQ kits combined with this NEB kit to make your cDNA. Okay, you're going to then use the SmartBell Express template prep kit 2 that we've talked about in the other lunch and learn sessions. It's that standard kit to prepare your libraries, and it's a less than a 1-day protocol, and you can multiplex probably up to 12 samples with barcoding and still get very high resolution data. Then you sequence on SQL or SQL2. This is for the latest models of the instrument, and just follow the protocols. Use the recommended settings for ISO-SEQ, and you'll expect to generate around that 4 million full-length non-categorized reads on a Smart Cell 8M. And then you've got a full data analysis solution built into the PacBio pipeline, so you can have this set with ISO-SEQ, and there's a few parameters that I mentioned before that you can tweak about what level of Q-score that you allow through, and various other things. You can up that Q-score. The example I gave was Q20, but you could up it to Q30 if you wanted to go to 99.9% accuracy, and that's what people are tending to do now, because the reads are so accurate. The filtering still doesn't remove many reads that are less than Q30, so that's great. So that's a sort of roundup of your best practices for running ISO-SEQ with PacBio Sequel 2. And just to finish off, it's really just a specialist, I suppose, application of ISO-SEQ, and but people are now running a single cell ISO-SEQ. So rather than capturing, whole transcriptome data, they're actually capturing specifically the transcriptome from a single cell, and there's a protocol for this because there's a number of single cell companies out there, such as 10x Genomics and others, that are capturing transcriptome information from single cells, from either dissociated tissues or from cell lines and all sorts of things, and they rely on a short read sequencing technology generally, and so they make full-length cDNA as part of their protocol, but then they fragment that down and only sequence a short part of it. But if you pull out of the, for example, 10x Genomics workflow, that we'll just give a quick example of here, if you pull out of that before the fragmentation step, you've got the full-length cDNA that's individually barcoded to be part of a single cell, so you know all the transcripts from together from a single cell. You can then put that onto the PacBio pipeline and sequence full-length cDNA. So there's a guide for this, as I say. It's a procedure and checklist, the usual PacBio protocol, but just when you're looking at these, be careful because the wordings are similar. So this is preparing single-cell ISO-SEQ libraries for SmartBuild template prep. So it's single-cell ISO-SEQ libraries, this particular protocol. So that's the one you want if you're interested in this. So it goes through all the lab guidelines, what you need, all the extra reagents, consumables, and things that you need, and instructions for performing the reamplification of your cDNA from these single-cell preps, and any other information that you need to grab those barcodes specifically to do the bioinformatics and things, and also then obviously making the libraries and everything else. So as I said, I guess you generate the full-length transcript isoforms, and these are assigned to individual cells because you've captured cells in some sort of, well, in 10x Genomics case, you capture them in their so-called GEMs, their gel beads in emulsion systems. So you capture a cell with a gel bead that's got lots of oligos, and those oligos are all have the same barcode. So they capture a single cell, and all the transcripts get barcoded with that same barcode to identify them. And you can then use your sequencing to identify those barcodes and demultiplex back to anything from an individual cell. So again, I won't read the slide to you, but you can always, if you're catching up on this, you can always freeze at this point and have a read through. But again, the same principles of ISO-SEQ apply. You'll end up with full-length transcripts with all the isoforms. There's no assembly or anything required. So there we go. So the Sequel system requires about 80 nanograms of cDNA, whereas for the Sequel 2 system for this, they requires about 160. So you've got a lot more reads on a single on the Smart Cell 8M as opposed to this 1M. So you need more starting material. Just something to note if you're doing this. So again, it's a very similar workflow once you get into the PacBio part as doing the standard ISO-SEQ. It's just that you're combining it with that starting single cell capture method of one sort or another. Yeah, we can run through how that works. I'm just conscious of time here. Sorry about the animations. Taking a little bit of a while to load. So this is really how you stitch the single cell capture part of the protocol together with the the PacBio sequencing part of the protocol through the guide that I showed you. And you'll see this page here looks very similar to an earlier slide from the standard ISO-SEQ workflow, where you had full-length messenger RNA, and you could capture the poly A tail with a poly DT primer. So it's a very similar sort of setup for there. And then you also the reverse transcriptase enzyme puts the 3 Cs on the end, and therefore you've got a template switch oligo that can capture onto that and do the second strand synthesis. So really, it's very, very similar. But built in to this method, if you're working with one of the single cell systems, is that this poly DT primer contains some special sequences in here, a cell barcode, and potentially a unique molecular identifier, which allows the mathematics to go on in the analysis pipeline, allows you to count the number of transcripts that you get per cell to work out the gene expression levels. So so this is included within the primer, just that little bit of extra magic, effectively, to make the cDNA with this detail from a single cell. And then the workflow runs essentially very similarly. So you do your first strand synthesis, template switch oligo allows you the second strand synthesis. You then have some handles on the end that you know about, so that you can do some amplification and the usual sort of cleanups. And at that point, effectively, you flick it away from a short read sequencing scenario that you might run from the single cell protocol from that sort of provider, whether it's 10x Genomics or others, and flick it into the PacBio's protocol. But at that stage, that you still have the full length cell barcoded cDNA. So there again, it goes really into a standard workflow. You have a molecule with its magic oligos on the end, and you can re-amplify to get enough. You do any damage repair and end repairs that you need, so that you're able to A-tail and put your SmartBell adapters on. From there, you end up with the SmartBell library that looks just like any other PacBio library, really, at that point, and onto the sequencer it goes. And therefore, if you're sequencing through that molecule, you're sequencing through the cDNA, but you're also sequencing those unique molecular identifier tags and the single cell barcode tags, which gives you the power to show which transcript comes from which cell and how many transcripts of a particular gene you get in each cell. And there's an example of that here in a publication from mouse, and a little sort of diagram cartoon of really how it was done. So this is from a mouse brain sample. So the poor old mouse gets hit on the head, and it gently hit on the head so it doesn't damage the brain too much. And they took cells from the cerebellum, and they made that full-length cDNA, but single-cell library prep. So this was actually using the 10x Genomics system, their Chromium controller, and running their gel beads and emulsion prep to grab single cells and barcode those transcripts. So it went through that standard protocol, through their Chromium controller instrument, microfluidics instrument to grab that. And then they took some of that library and went into their standard 10x pipeline, where they fragment that full-length cDNA and put the Illumina adapters and things on, and they did Illumina sequencing, short read sequencing to grab transcript information. But they also took the other half of that prep off the machine and took it into the PacBio ISO-SEQ that we described. So you get the various isoform expression. Software is able to identify those barcodes from each read and isoform and assign those to each cell type, and generate all that classification of barcodes and transcript, and put together this publication to show all the different isoforms in the mouse cerebellum sample. So again, the power of this actually gave them 2 lots of data. So they had the short read and long read sequencing data together to not only find all the transcript information, but also find all the novel isoforms. As you can see here, they found a lot of novel isoforms using this method that hadn't been shown before, novel splice variants and things that hadn't ever been identified before. So a powerful technique, and again, there's some best practices here for the amount of starting material. For sequel 2, you need about 160 nanograms of input cDNA, so hence the amplification steps at the very beginning, and that will allow you to run 1 or 2 SmartStyle 8Ms. Usual library prep with the Express Template Prep Kit 2. You can generate with this around about 3 million of the full-length reads, and to sequence those unique molecules. It's the standard setup for an ISO-SEQ run, 24-hour movie time, yeah, an idea of cost. This is US dollars in costs, so just a note there. And again, there's a specialized ISO-SEQ protocol and workflow to go through for the bioinformatics that's able to grab that's 10x in the case of the example I showed you, the 10x barcoding UNI information, but there are others systems that are also available that you can grab and characterize through the pipeline to do your analysis. So that's it for the transcriptomics talk, and also it for the PacBio Lunch and Learn sessions. Once again, apologies to anyone that tuned in a week ago and we weren't able to bring you the transcriptomics talk, hence it was brought on today. But hopefully this whole little idea of the Lunch and Learn sessions has been fun, has been informative for you, and we've tried to keep it a bit light and a bit more overview. We may decide to do something similar again another time, maybe going into more details about particular parts of the protocol or other applications, because we only covered, as I say, in 4 talks, 1 general overview and 3 different applications. There are other applications available for long read sequencing with PacBio. So thanks for your attention. I hope it's been useful. And if you have any questions, you're welcome to pop them in the chat or email me. I'm Paul Gooding at Millennium Science, so pgooding@mscience.com.au if you want any information. But otherwise, oh, there's a few things popping up in the chat. And what have we got here from Joseph? If you target 2K RNA size, what is the range of sizes obtained? Will it be skewed to smaller sizes? No, the distribution with the Pronex beads, Joseph, is fairly normalized. So if you're going for that sort of mode of 2KB, you're going to capture some stuff down to probably 1.2, 1.5, and up to maybe 3. So it's, you know, it's quite a well normalized distribution. Likewise with the other Pronex bead ratios, you're capturing a normal curve above your mode that you're after. Yeah, I hope that answers your question there. There's one in the chat, which is just a thank you. That's very nice. Thank you, Paula. So yes, unless there's any other questions or whatever, I will wish you a good day. Enjoy the rest of your afternoon, and thanks for your attention. Cheers.